I'm going to be explaining and drawing the membrane structure using the fluid mosaic model, which are made up of many different components. This membrane or structure rather is considered fluid because most of the components are able to move around, however rarely flip-flopping. And it's usually the proteins and phospholipids that are more fluid. Four important functions of the membrane are diffusion barrier, response to environment, substrate for reactions, and organization of metabolic pathways. The composition of the membrane is primarily 20 to 80% of phospholipid, making up 10 to 70% of proteins, which is specific to the mitochondrial inert membrane, 1 to 25% sterol, which can be found in certain patches of the membrane, oligosaccharides, which contain 0 to 5% weight of the outer membrane of animal cells. The phospholipid bilayer provides permeability barrier for matrix. Next, I'm going to speak about transmembrane protein, which is also known as the integral protein, which is what you're looking at here. Some compositions of the transmembrane protein is that it has carrying components which actively or passively transports molecules across the membrane, has channels which passively transports molecules across the membrane, and receptors that transmit information into the cell. The integral protein location is amongst the phospholipid bilayer, and it is largely hydrophobic, and it does not flip-flop across. However, it is lateral. The other protein is the peripheral protein, which is located at the surface of the phospholipid bilayer, and it is hydrophilic, and it bounds to integral proteins or the lipid, which is also the head of the phospho or phosphate group or tail. Some examples of the peripheral protein are integrins, which form the link between the extracellular matrix and the transmembrane proteins, spectrin, which links between the cytoskeleton, and G proteins, which are attached inside the membrane, and some detect hormone signaling. Here is the glycoprotein. As I mentioned before, it's attached inside the membrane, and some detect hormone signals. The peripheral protein does not flip-flop, but it is lateral. They immobilize the outer surface of the membrane, making it less soluble to small water molecules that could pass through.